Hello, hi everybody. Welcome back to today. Brief introduction class is about the protein purification and characterization techniques. You can see that slide by slide we will discuss about the protein purification and character. You can see the one of the basic slide I was kept here. The protein so mainly four kind of structures you can find out. You can see the primary structure, secondary structure, and tertiary structure and quaternary structure. So these are the main four types of structures in protein you will be see in depending on their characters. You can see here protein extraction and the purification. Protein extraction is the process of isolating and purification protein from samples of whole tissue, cell culture and biological fluids. And you can see the further four steps are needed to protein specially purification. One is the lysis, cell lysis, and second one is the protein binding to matrix and washing and elution. Based on these hydrophobic interaction, column chromatography, size exclusion chromatography, ion exchange chromatography and affinity chromatography. These are the separation techniques for depending on the various size, charge and various factors depending on the protein separations. You can see one of the best technique is the SDS polyacrylamide gel electrophoresis of protein. You can see SDS page is the most widely used for the quantitatively amazing analyzing the proteins mixture. You can see it is particularly useful for the monitoring the protein purification because that method is based on the separation of protein according to the size method. So SDS page gel is one of the separation techniques based on the protein size. It will be separate the our compounds or our mixture or our complex. So can also be used to determine the relatively mass of the protein. Then you can see here these are the few of the images how it is set up over the experiment of the SDS page gel. You can see that the electrodes and the cathodes you can use a positive and negative charge buffer, and high molecular weight proteins will be first and low molecular weight protein will move fast down from electrode to from lower to higher. You can see wells and large fragment and the smaller fragments will be settled down and it will come fast. You can see some of the images I was mentioned here. You can see, and you can see here this is the how is loading the image, and this is the control like a protein dye. This is one of the protein dye various molecular weight, and you can compare into the unknown our compound. This is the control like experiment. You can see this how you will supply that power to the SDS cell. So power supply you can see cathode and negative charge, and that anode is the positive charge. Depending on the protein will move from positive to negative charge depending on their size. And you can see the testing methods for the protein characterization. This is very important. I was mentioned here the flow chart protein identification and purification. These are the SDS page. Western blot techniques is one of the, these are the techniques to use to identify the protein, and the purification HPLC capillary electrophoresis size exclusion chromatography. Then charge isoforms is the gel capillary isoelectric fusing. Then after the whole column isoelectric fusing ion exchange column and HPLC. Next one is the aggregate state. You can see SDS page gel. Then after size exclusion HPLC light scattering. These are the protein aggregation states determination techniques or the identification techniques. Then after oxidation states HPLC with LC or UV, and reverse phase of LC enzymatic digestions. These are the techniques to protein characterization. Next one you can see Sandair, the specialty diagnostic private limited. These are the private one of the diagnostic techniques, diagnostic center. They are working in various applications, especially in protein chemistry. You can see here the polyacrylamide gel. They are using the negative gel and SDS page gel to protein purification. Then you can see isoelectric fusing. This is one of the techniques nowadays private sector and the diagnostic centers they are using. And you can see the two-dimensional gel electrophoresis 2D cell. Next one, high performance liquid chromatography HPLC. They are using for the purity checking. And the next one is fast liquid chromatography HFVLC for the purification of dissolved protein. And next one is the protein characterization using for the circular dichroism spectroscopy, whether how the protein structure are changing in the, especially in secondary structure, they can find by through the circular dichroism spectroscopy. Then after you can see MALDI, one of the finding of the best techniques for the mass identification of protein, exact mass analysis for the MALDI, matrix assistant laser dispositions of the ionization. Then after you can see GCMS, gas chromatography and mass spectroscopy, and then LCMS, liquid chromatography and mass spectroscopy techniques, and QTUF, quarter pole and time of flight. These are using for the exact mass analysis, protein filing. Then after it's the protein characterizations and sample preparation. These are the now plant samples, how we'll extract the protein, what are that, how to remove the excess amount of this concentration of protein. You can use these techniques. Animal tissues for the extraction of protein from various plants and animals, and dairy tissue cultures, others biological fluids they use for the buffer for the exert of hydrophobic and hydrophilic proteins. Then after you can see microbial sample, also nowadays they are sequential extractions of buffer using these things. Then you can see nutraceutical samples, also they are nowadays using protein purifications, ion exchange chromatography for separation, size exclusion chromatography techniques, and then amino acid compositions and peptide mapping, and the purity and by checking, and the impurity of profiling, and structure analysis. You can say this is very important, especially protein folds into primary, secondary, tertiary and quaternary structures for the activity and the stability. So the secondary and tertiary structures are the determined by the CD spectroscopy and fluorescence spectroscopy. These are the techniques important for the basics techniques to identify the protein structures. Then you can see the complete sequence of the various techniques: co-fast translation modification of analysis, quantitative proteomics. You can see emerging technologies, mass spectroscopy based on the proteins are used to powerful tool to quantitatively and systematically assess the quantitative difference in the protein profile, distinct samples and the increasing becoming the significant component. These are the based on the proteomics, 2D MALDI based biomarker they are using. And the next one you can see here, sample preparation of 2D. The important step to special care is to taken to the prepare the samples in 2D, since salts and buffers etc. Interface is sample migration in the first dimension near to perfect the sample protein quantitation will be ensured the reduce the gel to gel variations. Two-dimensional gel electrophoresis, 2D protein profiling using first IEFR on IPG trips to the second STS page, dimension of protein isoelectric points and the size respectively from 11, 7, 11 and 24 centimeter format of the gel to superior resolutions. Next one is the differential gel electrophoresis, two-dimensional gel electrophoresis. These are the one of the protein purification techniques. Next you can see the image analysis, how you will image analysis. You can see MALDI and digestions, whole protein stain band is cut, the digesting it trypsin. Trypsin is one of the protein will be mixed to the small parts, digestions. Next one say plasma amino acids. This is a biological techniques to number of amino acids are present in the blood. You can diagnostic techniques, and the glutamine is one of the most abundant free amino acids in human muscles, and the plasma is utilized to very high rates of eukaryotics to provide energy and optimal conditions for nucleotides biosynthesis. Then after you can see here various essential amino acids in their roles. You can see amino acids and their roles: isoleucine, leucine, valine, histidine, lysine, methionine, phenylalanine, threonine, tryptophan. These are the essential amino acid and their roles, what the importance. You can see here. Then after you can see here the color reactions of protein and amino acid identification. You can see here Biuret reactions. Violet are the purple color, then two peptide linkage will present. Ninhydrin reactions, blue color, then alpha amino acids are present. Xantho protein reactions. Orange color it will come aromatic amino acids, phenyl, tyrosine, tryptophan. You can see Millin's reactions, red color phenolic groups are present, tyrosines, and Hopkins Cole reactions, violet color indole ring is present, trypsin, okay, tryptophan. And I can see Saguchi reactions, intense red color, these groups are present, nitrosophuse reactions, and sulfur test, fuel in catalytic reactions. These are the one of the color reactions to the protein identifications. What are the amino acids are present? Amino acids are present, two peptide linkage, and how many amino acids are present, what type of amino acids are present into our mixture, you can find out through the color reactions of protein and amino acids. Then you can see here separation mechanism and techniques, property of protein and techniques, dimer of the molecule, size exclusion chromatography, electric charge of molecules, ion exchange chromatography, presence of hydrophobic domain, reverse phase chromatography, presence of hydrophobic groups, hydrophobic interaction chromatography, biospecific interaction, affinity chromatography. These are the properties of protein, and these are the what are the techniques we need to apply depending on the property of proteins. Next one, you can see the plants and their proteins. You can see here number of plants I was mentioned here, and the parts of the plant and the proteins. What are the proteins and pharmaceutical medicines application? You can see soya bean. So simply we'll call the glycine max. The seed part you can see B. Conglucin. This is the protein name. P, you can see here the nuts, peanuts, seeds, and you can see alpha and beta fistoids. These are the glycine. These are the globulin proteins are present various plants, and what are the parts and number of proteins, and these are the pharmaceutical and the medicinal uses. This is very important for real life applications. You need to read it thoroughly, these plants and their proteins. Then after, see what are the methods to extract initially for the protein from plants or any tissue, other animal tissues. CTA and acetone methods, phenol and SDS method. See, fresh the plant tissues, grind it L nitrogen, then after powder mix into the 10% C TCA, then after 0.00% of 2 any other solvent, vortex and sonication for the 5 minutes, then after incubate for the 1 hour at RT room temperature, and centrifuge the 9000 for the 20 minutes in 4 degrees Celsius. Then pellet will come out, then wash twice with other solvents, what is acetones and speed vacuum, then pellets and lysis buffer and protein. So these are the protein extraction methods. Then after you can see phenol SDS method. Just it will take the plant any tissues or plant anything and fresh it, grind it LNA2. Then protein extraction is phenol and SDS and methanol, ammonium acetate. Then after phenol protein precipitation, then wash, pellets and dry by speed vacuum, and then pellet, lysis the buffer, then protein. It will come the protein. These are the one of the techniques to plant to how extract the proteins. These are the two methods to plant to extract the proteins. Then after you can see here one of the protein diagnostic private limited in Bangalore, this the Raja Digview. He is the founder and director of this company protein. He is the earlier the IIS Bangalore scientist in biophysical chemist, and biophysical chemist he was work and various protein. Then he came out after retired, he came out and he established the protein design private limited company, and he now run is various factors depending on the diagnostic and therapeutic values development of protein based techniques. And thank you very much for your great patience. Subscribe, like, share and comment. Thanks for your kind attention for this listening of my class. So next class we will discuss about the detail about the protein purification